Thank you very much for inviting me here to present our work on de novo assembly and the phasing of dikaryotic genome. Puccinia graminis tritici is the fungus that causes stem-rust disease of wheat. It is a major threat to the global production of wheat. Rapid pathogen evolution has made disease control a challenging task. Therefore, understanding PGT population dynamics is critical for devising effective disease management strategies. PGT requires two hosts to complete its life cycle. New genetic variants of PGT can arise from both sexual and asexual mechanisms. Barbary serves as the initial host where sexual cycle of the fungus is completed. Spores from Barbary can infect the primary host wheat. PGT produces 5 spores in different life states. PGT completes its asexual life cycle on primary host, in this case, wheat. Spores is produced in wheat and can reinfect, and it has ability to spread from plant to plant. Spores is a haploid which can infect Barbary. During spores, fertilization occurs when a spore meets a receptive hyphae. Spores such as spores, telospores, and spores are dikaryotic because they have 2 nuclei per cell. PGT isolate 18SPA92-1 was recovered from stem-rust samples collected in 2018 from a wheat field adjacent to Barbary plantations in Spain. This isolate was typed as RES TKHBK with the wheat stem-rust differential set. It produced high infection types on several genes such as SR5, SR6, and SR9 alleles and other genes. It also exhibited virulence on lines carrying SR53, SR53, and SR59. From this unique virulence and evirulence, it is hypothesized that this isolate, 18SPA92-1, is a sexually derived race. The objectives of this study were: 1, to create a genome assembly of 18SPA92-1. 2, to compare assemblies, gene and the protein predictions with genomic resources of asexual PGT population. And also to determine genetic relationship between 18SPA92-1 and other PGT isolates. DNA extraction was done from 1.5 gram of spores. After quantity and the quality of DNA were checked, sequencing was done at University of Minnesota Genomics Center. Whole genome sequencing was conducted using PacBio high-fidelity sequencing technology. Genome assembly of isolate 18SPA92-1 was built from PacBio high-fi reads using de Novo assembler. Reads with lengths less than 15 kb and the quality value less than 20 were filtered out. And approximately 50x coverage was used for the genome assembly. To remove contigs of plant organelles such as mitochondrial, ribosomal, and chloroplast DNA, local NCBI nucleotides database was used. To assess the completeness of the genome assembly, Busco genes was used. Repeat contents and the presence of telomere were checked. Both primary and alternative assemblies of the 18SPA92-1 were annotated using the fan-annotate pipeline, which included 4 prediction software programs. Interproscan 5 was used for protein function prediction. Phylogenetic analysis was conducted using both Almuna DNA sequences and RNA-seq data downloaded for global PGT isolate from public databases. Trimmed reads were aligned against indexed 18SPA92-1 assembly using ISA2 software. Phylogenetic tree was constructed in Tussle software. Assembly basis statics is shown for 2 assemblies in this table. The primary assembly consists 95 contigs, whereas the alternative assembly has 93 contigs. N50 of primary assembly was 4.6 megabase pair, whereas alternative assembly N50 was 1.6 megabase pair. The haploid total length for primary assembly was 98.7, whereas 76.5 for alternative assembly. Both primary and the secondary assemblies of 18SPA92-1 comprised over 94% of conserved fungal Busco genes. This shows that the assemblies are complete. All 18 chromosomes were recovered for this assembly. 15 of 18 chromosomes were represented by a single contig. Chromosome 7, chromosome 9, and the chromosome 17 were represented by 2 contigs, or more contigs, I mean. Chromosomes vary in length. Except chromosome 3 and 8, all chromosomes contain telomeres based on the primary haplotype assembly. Structural evaluation of 18SPA92-1 was determined using haplotics in the PG21-0 assembly. In terms of number, insertion and the deletions are large in numbers than others. But in terms of size, repeat contraction is larger than the others. Overall, the size of structure variation was about 9% of the primary assembly size. PGT21-0 assembly is the high quality published assembly available so far. At chromosome assembly level, there's a good collinearity between corresponding chromosomes of PGT21-0 and 18SPA92-1, except at chromosome 3 and the chromosome 6, where inversions were detected. Repeat sequence content was also determined for this isolate. Generally, about 50% of the assembly is repeated sequence from different classes of repeat classes. Interspersed repeat class is larger than the other repeat classes. A total of 15,017 genes were predicted for this assembly. And also proteins predicted for this assembly, and a total of 14,192 chromosomes proteins were predicted. Out of this, 1,215 proteins are secreted proteins. This circus plot shows the distribution of genomic features on chromosomes. The outer track is for number of genes. The middle one is for number of SNPs, and the inner track is for structural variations. So the way this was done is the number of genes per 100 kb was counted for each chromosome, and also the same for SNPs and structural variation. So the zigzag shows the variation across different regions. Phylogenetic analysis was conducted using 32,390 SNPs, which are identified based on 34 global PGT isolates. The 18SPA92-1 was indicated by red arrow on the top right. It is closer to isolate from Europe than PGT21- from South Africa, where PGT21-0 was clustered with other races from South Africa. Similarly, ETKSK, which is the original UG99, it was clustered with other two races from Ethiopia, ET01 and ET02. So 18SPA92-1 related to IR01 with similar geographic origin, that is West Europe. In summary, haplotype resolved assembly was developed for one sexually derived isolate of PGT. The assembly is comparable in size and the quality with previously released PGT assemblies. The 18SPA92-1 chromosome assemblies were largely collinear with the PGT21-0 chromosomes, with exception of chromosome 3. Number of genes and the proteins are in the range of that of other isolates. From phylogenetic analysis of global PGT races, isolate from the same geographic origin were clustered together. At this time, 3 more isolates have been submitted for sequencing. Finally, I want to acknowledge Eugene Lab, particularly Radhika and Pablo. Radhika played significant role in DNA extraction and working on sequencing data as well. And also Pablo contributed to this project by phenotyping the races. Majority of the lab work was done in Zabo's lab, and also some lab work was done in Rose lab as well. The fund for my research came from USDRS National Plant Disease Recovery System. With that, thank you very much for listening. If you have questions and comments, you are welcome.